works. I've been advertising this for quite some time, but today is the day. So let me remind you that we talked last time about some examples of quantum effects in biology, and the crown jewel in this area is photosynthesis, which has been demonstrated to operate on quantum principles. I also mentioned metabolism, and in a strange way, both of these effects have consequences for microtubules that you see here on this slide, and you see them here on the left in an interface of a dividing cell, and here microtubules in a mitotic cell forming mitotic spindles. Actually, it's not a single microtubule. It varies, but in the human body, it's about 30 microtubules forming one spindle and segregating the chromosomes, applying a large amount of force. But of course, this is only one function of microtubules. There are many others, including transport and motility to some degree. But this is basically dividing cells, and in non-dividing cells, in particular neurons, there is a lot of evidence, direct and indirect, of the microtubule cytoskeleton. They form a network with other proteins, such as microtubule-associated proteins, spanning the neuron's length. And in the axon, as you can see here, you have microtubules interconnected by MAPs. So microtubule-associated protein, we will call it MAP for short. MAP tau, MAP2. There's a whole family of these proteins, and they allow microtubules to form a scaffold inside neurons, and it's well known that this is stabilizing the morphology of neurons, but it's more than that. There's also very robust transport taking place, exoplasmic transport on microtubules with micromotor proteins, and especially interesting is the role, somewhat enigmatic, but at least indirectly implicated in cognitive functions in dendrites, where there is a unique orientation of microtubules, which is antiparallel. That is, microtubules have plus and minus ends. This is from the point of view of dynamics. So plus ends are more dynamically growing, minus are shortening typically. That's a separate lecture on the polymerization of microtubules. We have no time for that. But in dendrites, it's pluses and minuses alternating with the neighboring microtubules, and the suspicion is because motor proteins, as some of you may know, move unidirectionally in a processive motion. So kinesins all move on microtubules to the plus end, dyning to the minus end. And when you have alternating orientation of microtubules, you can have feedback loops and perhaps information transfer back to the nucleus or the centriole, which we briefly discussed, maybe the command center in all cells. In this slide, it also mentions, it's mentioned that learning and memory involves reordering of the microtubule cytoskeleton, reorganization, and strengthening of synapses, which is called long-term potentiation. But almost all, if not all, cognitive diseases, neurological disorders are related to the deterioration of some form of microtubule cytoskeleton and the related proteins that involves Alzheimer's disease, various dementias, bipolar disorder, Huntington disease, schizophrenia, and traumatic brain injury as well. In traumatic brain injuries, microtubules get broken in neurons, and then that causes cognitive dysfunction. So that's still kind of on a classical level, but I want to now relate it to what we were talking about before, namely tubulin, the building block of microtubules, has a network of tryptophan residues, tryptophans, and there's 8 of them in each tubulin dimer. This is a tubulin dimer, beta alpha monomers forming a stable dimer, which is a building block of the microtubule surface. And then it falls into a cylinder. And these tryptophan networks are drawn here as pathways for potentially independent communication channels. In fact, electronic, possibly, because tryptophans can be photoexcited, and electrons and tryptophans can be transferred to a conduction band. And that's number one. And number two is, as you will see next, a similarity between FMO complexes in photosynthetic systems that we talked about as the primary site of quantum excitations in leaves and plants and bacteria, where photon energy is absorbed, transmitted, and used for various purposes, structure formation and energy production. And this is very similar. I've shown you a similar representation for the FMO system. Here is the network of 8 tryptophans. The photosynthetic 8, right? So 8, and so that's the same network in a single tubulin. Here is the protein structure, and this is just abstracted out, showing you these aromatic rings here, and they're very detailed distances and orientations produced from our computer simulations with my former student Travis Craddock. That was part of his PhD thesis, by the way. He's now a professor of biophysics at the University of Waterloo, and he did some groundbreaking work here, and later on I'll tell you about additional work he's done at the University of Alberta. So that's just a sort of structural similarity, suggestive, and we've done simulations with Travis to demonstrate that these can be quantum excitations which are shared between the 8 groups here, and later on I'll tell you about the experiments which demonstrated the correctness of this prediction with some new information obtained. This is the dipole moment that each tryptophan residue possesses, and these dipole moments interact. It's a dipole-dipole interaction, and this is something maybe that you may not be so interested in seeing, but just to show you that this is pretty, I would say, well-defined physics problem with the Hamiltonian, which is a quantum representation of the energy energy of the system with interactions. This is the diagonal terms, and this is the interactions, dipole-dipole interactions. And we did all these calculations and published the work. This is again Travis Craddock's work, and the decay of these excitations over picosecond timescales were predicted, which is very, very short, but nonetheless comparable to photosynthetic energy capture, photons. Remember, we talked about the photoelectric effect in one of the first lectures as the key, one of the key experiments performed in the early 20th century, and Einstein explained it with the concept of quantization of the photon energy, h times f, frequency times Planck constant. And that was huge, a huge discovery that demonstrated the correctness of Max Planck's predictions. And in addition to the black body radiation, here we have something biologically analogous. Photosynthesis first, we saw and again demonstrated in about 15, 20 years ago, experimentally and conceptually illustrated by modeling. This takes it to the next system of great importance in cell biology, microtubules. It's a prediction at this moment that microtubules may be able to absorb electromagnetic quantum energy in the form of photons and collectively distribute a wave function throughout this network, and maybe also beyond. If you can imagine that there is a whole lattice of these tubulin dimers, and perhaps they interact and form a global quantum state for the entire microtubule, which was hypothesized, but never really experimentally demonstrated yet. The work with Aristide Professor Dogariu from University of Central Florida on delayed luminescence showed something leading to this conclusion experimentally by illumination of tubulin and microtubules in a solution over periods of about 2 minutes, interrupted for about I believe 10 minutes, and then illuminated and so on and so forth, and then with laser light of about, I believe, 630 nanometer wavelength, and then the measurements were made on the re-emitted light from the system. That's the experimental setup. Again, very complex optics. 630 nanometer wavelength. This is the corresponding energy, about 2 electron volts. For the physicists, that's maybe making a lot of sense. And by the way, the intensity of light that was used is not very large. It's comparable to the light intensity when we are exposed to sunlight. So it's not very unusual. And these are the results, which were very interesting because the time it took to re-emit the light from the biological structures, namely microtubules and tubulin, several experiments were made with different, this is in solution, ionic solution, with different concentrations, and tubulin alone in solution versus microtubules, or combinations here. At any rate, in all cases, you can see the intensity of the emitted light is very high initially, over fractions of a second, in fact. This is maybe not to scale. And then what's amazing is that it persists for minutes. And that's puzzling. We published results with just recently with maybe a refined setup and repeated experiments. Perhaps minutes is a little bit of a stretch, but definitely many seconds. And why this is important? Remember, in the beginning of this lecture, I mentioned the role of microtubules in neurons and in nerve cells in our brain. And if there is optical signal production and communication, the whole area of neuroscience needs to be revised and understood from scratch. And furthermore, microtubules may be actually quantum devices that operate on a time scale, at least, which is easily reconcilable with physiological effects. We have reaction times. I think I mentioned that was measured by Libet and neuroscientists I believe in the 60s, to be something like 500 milliseconds. Half a second is the reaction time. So these kinds of orders of magnitude seconds, at first thought to be incompatible with quantum systems, which don't have states that last that long. But now, if this is the argument presented in favor of quantum states in the brain, then the time scale probably is not a problem. This is another illustration of this effect here. And again, you have minutes, and this is the intensity of re-emitted light. There are, of course, some qualifications because these are microtubules in solution. That's not really inside neurons. Nobody has done such experiments in living neurons yet. And by the way, if anybody could do this and demonstrate, that would be, I don't know if this is a Nobel Prize winning measurement, but something really very, very important to communicate if it was done, but it so far hasn't. So I mentioned microtubules also in the context of metabolism, because microtubules are structures on which mitochondria and cells move, involving kinesins as the motors that carry mitochondria, but they are moving on microtubules that form the let's say roadways inside the cell. So everything is connected to energy production, mitochondria, and by the way, this may also explain. If somebody asked the question, oh yes, you've done these experiments in the lab with laser light, and you demonstrated that microtubules re-emit light after many seconds. And that could be used for some sort of information processing, optical information processing. But where is the light coming from? That's the big question, which is called biophotons, right? Where do biophotons come from? Definitely not from lasers. We don't have any lasers in our brains. But mitochondria are energy-producing organelles, and as a byproduct, they emit photons, electromagnetic waves, principally in the near-infrared range, so a bit longer wavelengths than the ones that we used in the experiment, something about 1,000 to 100 and 1,100 nanometers, as opposed to 630, but not exclusively. And there are some, technical aspects that need to be worked out, but it's definitely a question of feasibility has been addressed in the affirmative. Now let's zoom out a little bit and look at the brain questions from the point of view of quantum biology. So there are big questions, and here you have some sort of illustration of the organization. It's a synaptic connection, and microtubules will be way smaller than this inside dendrites and axons, as you saw. Here we have neurons, and so this is where microtubules will be forming these networks. So and then you have the whole brain with about 20 functional domains and lots and lots of neurons. You'll see in a second how many and how many connections. So these synaptic connections. So that's one of the most complex biological structures you can find on this planet. And these are big questions, maybe the biggest question, the largest, the most profound questions in all of science, although they have been skirted around for centuries, never really addressed head on. So in science, in philosophy, maybe, but what is consciousness? Where is memory stored? Where in the brain is memory? We don't know. And then what is the computational power of the brain? Namely, how many bits per second can it process in its maximum efficiency performance? And then what we are trying to address here is information processing in the brain classical, quantum, or maybe somewhere else. An answer is given, fractal, resonant. There are various proposals in the literature. And then we'll quickly look at also some of the maybe less profound but equally important questions, like how can the brain work with so low power, such a low power level compared to computers, as you'll see. It's an enigma. It has to do something way better than our computer engineers can today. So there were a lot of conferences over the past almost 3 decades, entitled "The Science of Consciousness" or "Toward the Science of Consciousness." Originally, they were and still are actually. The next one is coming up in July. It'll be held in Barcelona, Spain. But every other year, they're held in Tucson, Arizona, in the USA, and organized by Stuart Hameroff, who is an anesthesiologist at the University of Arizona. And I participated in many of these, not all, but they are fascinating meeting places for people with a deep interest in the area of consciousness from various angles, including spirituality. And there were some plenary sessions on East meets West, how Eastern spirituality religions can shed light on consciousness, and how can they communicate with the Western philosophy and scientific approaches. So this was 2 years ago in Taormina, Sicily, in Italy. Beautiful place. Been there, but not at the conference. And as I said, this year is Barcelona. So these conferences kind of gather people who believe in a, not only believe, but work on a scientific approach to this topic. Some people dispute it very vigorously. Robert Epstein here posted on some website, claimed that the brain is empty, it doesn't process information, retrieve knowledge, or store memories. Brain is not a computer. Of course, it's not a computer, but I would say performs computation, among other things. And some of these arguments are honestly ridiculous. Like, you will never find a copy of Beethoven's Fifth Symphony in the brain. Of course not. Copies of words. I mean, this is beyond ridiculous. And you know, some questions, of course, are legitimate, but this is not. So let's come down to earth on this and try to address some of it from a physics point of view. So here are some numbers I want you to keep in mind, pun intended. So there's about 100 billion neurons in the brain. To be specific, I don't know, well, computing this number is more of an estimate than a real bean counting, but it's somewhere on the order of 97 billion neurons, let's say 100 billion, which is 10 times roughly or more, 12 times the population of the planet Earth. So if you were to think about each neuron as a person, then it's a very crowded system. And there are, of course, other cells in the brain, not just neurons. There are glial cells and astrocytes and so on, pyramidal cells. But that number is worth keeping in mind. And then there is, on average, about 10,000 synapses per neuron. In other words, each neuron interacts directly with another 10 to the fourth neurons, 10,000 neurons. Some of them more, up to 100,000 maybe, and some less, but that's the average. And so imagine how information and signaling dense this system is. 10 times as many neurons as people on the planet, and each neuron person, let's say, communicates with 10,000 other nonstop. With the current technology, of course, you know, some people have millions of fans and followers on various social networks, but on average, I don't think we have 10,000 even internet friends or Facebook friends. At any rate, so that's kind of to give you a perspective on the numbers. So that gives us the number of 10 to the 15th synapses in the entire brain, and even conservatively counting the number of signals or impulses or action potentials, if you will, that are hallmarks of the neuronal activity, 10 impulses per second gives you an astronomical number of computing operations, if you will, 10 to the 16th every second. Now, this is comparable, not comparable, but 10 times greater, conservatively speaking, could be much more, as you will see later, than what one of the supercomputers, one of the most powerful supercomputers can do today. It's called Blue Gene's, IBM's Blue Gene computer or supercomputer with hundreds of thousands of processors. This is the one that beat Kasparov at chess, but yeah, has 10 to the 15th operation, so 10 times less, or maybe more, less than that even. So, okay, that's reassuring. Our brain is very powerful, can do a lot of operations per second. Maybe we don't use all of that, but that's another question. But what is really astounding is that it can perform these many operations at a minuscule power consumption rate, namely 25 watts, which is about 25% of the human body's energy demand per unit of time. So 25 joules per second. And that's the demand that is equivalent to what a night lamp on your by your bed is using, and that's our brain's consumption rate. You compare it to the Blue Gene, which requires 1.5 megawatts of power, which is, roughly speaking, 100,000 times more, and doing 10 times less. So it's a factor of, let's say, 1 million in orders of magnitude between the brain and the best supercomputer. The gap may be shrinking, but okay, we are still far apart. So these are the questions. And by the way, one of the possible answers to the question how it can be done with such low power demand is that if the brain operates as a quantum computer, then information processing is much cheaper when you do it in a quantum way. There's something called the Landauer theorem in classic information theory that tells us that each bit of information costs about kT times a kT of energy, which is K is the Boltzmann constant, T is the temperature. That's about 25 millivolts, milli-electron volts of energy to erase one bit of information. And so you can do the math, and that would be violated with this many operations. However, if it's at least part of it, or a large part of it, is performing quantum computation, then this theorem no longer applies, and instead of the so-called amplitude, which is, let's say, 1 for each bit, we can use the phase. I think I explained it in one of the lectures. Phase doing the computing phase interactions in a manner which is, for example, involved in the Josephson junctions and superconductivity. That was a Nobel Prize winner, Brian Josephson, who discovered this effect. Then this basically becomes almost free, energetically speaking. All right. So there are some ideas. If quantum effects play a role in cognitive functions of the brain, then, for example, what we talked about as the collapse of the wave function, quantum wave function, could be understood as a realized thought. So before we realize a thought, let's say make a decision about what to order in a restaurant, and you look at the menu, and there are several possibilities, all of which are superimposed as some ideas in your brain. Like, is it a pizza or a hamburger or a spaghetti or something? then eventually you make a decision, and that will be this quantum mechanical collapse of the wave function when you actually make this experiment and you really have to say what you want. Okay, and then if with quantum mechanical approaches to the processing of information in the brain, there might be also some limits on the predictability of our brains. There are some efforts in the US principally to read our minds basically by looking at the brain waves, doing the EEG or fMRI or something like this, and decoding it with maybe AI and trying to guess what we are thinking about. But there could be really severe limitations if we are using quantum mechanics in our mental processes, because there's this uncertainty principle and probabilistic approach. This would also have major implications for artificial intelligence, which is, at least today, performed in terms of classical ideas, not quantum, and that changes everything if artificial intelligence algorithms utilize quantum computing. Okay, so a lot of these discussions originate actually about consciousness, the brain, quantum neuroscience, let's say, originate with this book, The Emperor's New Mind, by Sir Roger Penrose. He is a professor at emeritus, I believe, at the University of Oxford, and recipient of the 2020 Nobel Prize in Physics. So very respectable, in fact, maybe one of the most famous scientists alive. He received the Nobel Prize for something else, for his contributions to cosmology, mainly, Nobel Prize in Physics. But nonetheless, he spent this was, I believe, in the late 1990s, so the last 20 plus years, thinking and publishing on this topic. So we shouldn't dismiss it. And in this book in particular, he emphasized the need for quantum physics, or even something that we don't currently even have, something more sophisticated than quantum physics, probably, but definitely not classical physics, to be able to deconstruct or understand how the brain works. And that started, in fact, a whole area of investigations. And together with Stuart Hameroff, I mentioned before, who is an anesthesiologist, Penrose and Hameroff propose a quantum model of how the brain may function at the level which is orders of magnitude more, I would say, precise than what current neuroscience considers. So currently neuroscience is basically looking at synaptic connections and activation and deactivation of electrical signals called action potentials, which is basically the flow of ions. What Hameroff and Penrose proposed in around 1998, I believe, was the first paper, something like that, maybe 7. So we are now talking about more than 25 years ago, still publishing and still refining their model. But the basic idea is still the same, namely, and you see here a neuron, in fact, a dendrite here, and a synaptic connection, and here's a single microtubule, of which there are many inside in parallel bundles, interconnected by the maps that I mentioned to you in the beginning of this lecture. But what's important is that they claim that each of these building blocks, namely tubulin dimers, is a quantum computational unit, which we call the qubit, as opposed to the classical unit being a bit. And why is that? Because it has a quantum wave function, and it can be in a superposition of two states or more, perhaps. And they wrote many papers, and the gist of it is that there is, in addition to this being a quantum state, it's also a state which self collapses or undergoes this transition to or a measurement process, if you will. So by gravitational interactions between two states in a superposition. It's a little bit, I would say, wild and weird, because it combines gravity with quantum physics, which, as I mentioned to you in the first lecture, I think nobody has done before. There's really no quantum gravity theory that is acceptable today. So combines two things which are still irreconcilable to explain something which is at the moment hypothetical. Okay, so, but nonetheless, that spawned a huge amount of interest, many papers, many scientific discussions. If you ask me, it's plausible, but there are some big technical problems to accept as is. So in my opinion, what the main challenge is how to connect the various levels of organization, as you can see here from a tubular dimer, even if we accept, as I argued, that it's actually now experimentally plausible that there is a quantum state here, and then how does this state extend to a whole microtubule, and from a single microtubule, how it entangles itself with other microtubules, let's say here in the dendrite or axon, and beyond that, how do you create a quantum state for the neuron, and then how do you create a quantum state for a group of neurons, and on, and then for the whole brain. So connecting the different scales of organization in a consistent way is non-trivial, to say the least. We know a lot about signal propagation, so that's kind of standard neuroscience approach. But I think it's while this is very well documented and understood, it doesn't go deep enough, because it simply considers each neuron as a binary switch, off and on, just like a light switch at the entrance of this classroom. And then you have just flipping of switches that are supposed to be giving some sort of computing power. Maybe computationally you could even accept it, but from the point of view of creating consciousness, for sure, or some cognitive ability, I find it grossly inadequate. Okay, so we know that microtubules play major roles in it, so I accept that, including how they interact with ion channels. So interactions of through the membrane between the cell and its environment, including neurons, can be understood. And if this is quantum, then it's even more interesting, because some of these interactions could be probabilistic, as we know they are. And then inside the dendrite, you have these networks of microtubules. And I want to point to your attention to the so-called C termini. So that's a microtubule here in green, and actually blue and green. And here's another microtubule, and they are connected by maps. But these little matchstick-like objects at various angles are carboxy terminals, C termini, and they are very elastic. They are full of electric charge, and they act basically as antennae that are sensitive to electromagnetic fields, including light, photons. So this is where action might be. We don't know yet, but that would be a very strong possibility. And all of it is dynamic, which, by the way, the OR theory of Penrose and Hameroff is not. In fact, there is building and rebuilding of these things taking place on different time scales, and also transport of material, including mRNA and neurotransmitter components and so on. So yeah, a static model doesn't cut, but yeah, as I mentioned earlier, dynamic and in fact self-healing as well. So there may be some hope for future therapies for these terrible diseases, which are all related to distraction or impairment of the microtubule cytoskeleton. So how to reconstruct, how to repair, how to strengthen them should be the focus of attention in the therapeutic area. So this is what started the idea with microtubules being some kind of computing device. This is from an earlier paper by Hameroff and Watt. Prior to Penrose, they looked at classical versions of information processing take place in microtubules. These are so-called cellular automata. And some of you who are more in the math, physics, information science areas are familiar with cellular automata, which are very simple but powerful models of computation involving just patterns interacting in the neighborhood with some simple rules, cellular automata. And Stephen Wolfram is one of the creators of this field, and he believes that all of physics can be simulated by these kinds of models. So if you can simulate all of physics, maybe you can simulate all of the information processing in the brain. But as I told you, I think it really is not possible because of the quantum systems. To my knowledge, well, there are some quantum cellular automata, but this wasn't the case here. Okay, so if you look at strictly speaking, I just want to point to some, again, numbers, because I'm a physicist, so I like numbers. There could be many channels of information processing and information storage, or in other words, memory formation, that microtubules possess. I mentioned to you C-termini that can have at least 4 states per dimer, because there are 2 C-termini that could be in the stretched or bent state. There could be electron hopping, let's say between tryptophans, at least 4 per dimer. There could be also, we know that they are actually, not could be, conformational changes which are related to GTP hydrolysis on beta tubulin, and so on and so forth. So a minimum of 32 states per dimer would give you about 100 kilobits of information per microtubule if you calculate the number of dimers in the microtubule of, let's say, 10 micron length on average, and then 1 gigabyte of information storage per neuron. And with 100 billion neurons, you could store 10 to the 20th bits of information per brain. For your information, you can buy a memory stick. In the olden days, it was several gigabytes. Now it's terabytes. You can buy terabytes. Tera is 10 to the 12th. But this is 10 to the 20th per brain. So yeah, so you have 8 orders of magnitude gap between a memory, state-of-the-art memory storage, memory stick of today. So what is it? 100 million times. And there's also the speed of transitions that can occur. That's in principle, if they are taking place on a nanosecond time scale, which is, let's say, compatible with different states of the protein, or even microseconds, 10 to the 6 transitions per second. So you would have something on 10 to the 26 or 10 to the 28, which is just unimaginably large number of information processes taking place per second. I will skip this. This is just to tell you what the limitations might be in terms of because these can be, if classical, they require energy supply, which can be coming from mitochondria. But nonetheless, even if you restrict this to smaller numbers due to this, you still end up with very, very impressive possibilities. So that's kind of a very conservative estimate with classical computing, 10 to the 20 bits per second per brain. Okay, now I will continue this story with with a little bit of a discussion about the memory. Where do we store our memories is an open question. Memory is actually not just storing, but also recalling information. Very importantly, otherwise there would be no use for it. And currently, the paradigm is based on the so-called long-term potentiation theory. That means that synapses, which are frequently used, let's say you're studying a subject or a language, or you're learning how to play a game, tennis, or you study German, then different areas of the brain are used, and these areas grow stronger connections between the synapses of the neurons in this area, and this is how long-term potentiation works in terms of making memories. I think it doesn't really scratch the surface, because also there are some neurological and physiological documented effects on recovering memories, or retraining different brain domains for memory storage and other functions following accidents or injury and so on. So that there are some major problems with this. But before I propose something alternative, I want to say that various people, famous people, looked at and tried to compute the amount of memory that can be stored in the human brain. And in this 1970s, anatomists were proposing that the brain can have up to 10 to the 16th synaptic operations per second, which is not too bad. So fast processing, almost close to what we mentioned here, a bit higher, 10 to the 18th and the 20th. Thomas Landauer, in this paper from 1986, estimated the memory storage capability of the human brain by saying that we retain only 2 bits per second of visual, verbal, tactile, musical memory, which I think is really ridiculously small, which would end up with something like a gigabyte or gigabit of memory that is even less than an old memory stick, somewhat depressingly. Yeah, so that's the calculation, 2 bits per second, and a human lifetime in seconds is 2.5 billion, so something on this order. I definitely disagree with this. And in fact, John von Neumann, who is, as you know, the godfather of computing, modern computing, in 1950 estimated what we are claiming now is actually very close to a reasonable assumption estimate, 3 times 10 to the 20th bits. So that was what, 75 years ago. And I'll show you, well, I showed you before that this can be the case when you look at all the possible modes of memory storage. I think I missed it, sorry. Where is that? Here, yes. Yes, so here in this approximation, you have 10 to the 20th bits per brain, and this is the same order of magnitude as John von Neumann predicted. Okay, so some other people, scientists, cast various doubts on this kind of approach to memory storage. Famously, Karl Pribram, a very well-known neurophysiologist, proposed a holographic memory production in the brain, so three-dimensional images produced by biophotons. Rupert Sheldrake, who is a popular science writer, some of you might have read his books, some videos, YouTube videos you can find quite interesting. This is far out. He's claiming that memories are not even stored in the brain at all, but they exist in some form of ether or aura or field, memory field. That's why he called it what's it called it? I forget the name. Sorry. Maybe I'll come back. All right. Anyway, there's a memory field outside of the human body. And then there were some very interesting experiments performed by Michael Levin from Tufts University in Boston, who actually worked with planaria, with worms, then trained them to solve some puzzles and mazes, just like mice are usually trained. And then he decapitated these poor worms. They survive and then regrow their brain, their heads, with tiny little brains. And apparently they were able to solve these puzzles in spite of having lost the brains that were supposed to store the memory. So there is something interesting. Perhaps memories are not just in the brain, but elsewhere in the body, or maybe even outside. I don't really subscribe to this point of view of Sheldrake, but I find it very interesting. So with Travis Craddock, we worked and published some interesting alternative theory of where memory can be stored, and not based on long-term potentiation, but it's based on the cytoskeleton, in particular the microtubules. And it was actually initiated by experiments performed by American scientists who inhibited a particular enzyme called calmodulin kinase 2, CAMK2 for short, in mice. And these mice, with the inhibitor administered to them, they lost memory. And in this case, they were not able to solve mazes anymore. I believe this is a reversible process. So what's happening? So, of course, a synapse is involved in it, in particular calcium waves or calcium activation of these enzymes. So the enzymes are on the postsynaptic side, and calcium travels across the synapse and activates the enzyme, which then, through various steps, which we don't need to go through in detail, phosphorylates microtubules. And that's the connection. We published with Travis again, and Stuart Hameroff was directly involved in the project. We published a paper that not only explained how this might work, but produced computational models of very precise interaction between this enzyme and microtubules. So here it is, CAMK2 phosphorylates. So it leaves a phosphate group on either a serine or a threonine residues. This is a distribution of this serines and threonines on the microtubule surface. This is a charge representation, electrostatic charge representation of a tubulin dimer. This is the same tubulin dimer without the charge painted on it. Blue means or represents a positive charge, red negative charge. And you can see here how a patch of a microtubule like this electrostatically looks like that. Or there are actually two lattices. So you see here more rectangular here and more hexagonal. This is depending on which lattice is involved. And this is the same patch painted electrostatically. Why am I saying this? Because as you will see, there's an electrostatic attraction between this CAMK2 holoenzymes and tubulin. Okay, here it is. So Travis has computationally reconstructed this calmodulin kinase 2 enzyme, and it looks, this is the view from above in the top layer, top panel, and it looks like a snowflake, let's say, beautiful snowflake, hexagonal. And here's the same snowflake painted electrostatically with the same color code as before. This is so that's from the top is from above, and this is sideways here. The same enzyme seen sideways, and again you see now that it has sort of legs, and there are various metaphors for it. I think Stuart Hameroff calls it the Terminator or nanopoodle, because this is nanometer dimensions, and it basically moves around along a microtubule like so, in this direction, stepping on tubulin dimers and phosphorylating them. So phosphorylation, as you probably know, is one of the major signaling processes in all of biology. So activates a protein, for example. So first of all, we looked at matching geometrically these structures. This is the enzyme from above again, and this is the surface of a microtubule from above. It's either hexagonal or rectangular, and this is superposition of these snowflakes or enzymes on these surfaces, and you can see the match is almost perfect. So that's an argument in favor of some kind of strong interaction, but it gets better. Let's see, I don't have a picture here, but it's also matching electrostatically, which means that these blue areas here, blue areas you can see match the red areas on tubulin, and vice versa. You can find all of these images plus explanations in our paper from 2012. And we also use this to calculate the amount of information that can be stored in microtubules this way by phosphorylation. And because it's a dimer, so you actually can have three states. You can have phosphorylation of the top monomer, phosphorylation of the bottom monomer, or an unphosphorylated state. So three states, so we call it not bits but trits, and then we calculate how many such states can be encoded in the microtubule. And it turns out it's about 1 gigabyte per neuron, as we calculated before from other means. So it's very compatible. And then of course you do the multiplication by the number of neurons. So this is gigabyte is 10 to the 9th bytes, and there's 10 to the 11th neurons. So you multiply these two numbers, so plus times 10 to the 9th times 10 to the 11th gives you 10 to the 20th again. Remember John von Neumann, 3 times 10 to the 20th. This might be probably the best candidate yet for optimized modeling of memory storage at the molecular level. Each element here that can be encoded with 1 bit. In fact, the phosphate group can be a quantum state. So I don't want to push too far with this because it hasn't been published or analyzed in detail, but the phosphate group is possibly bound or unbound or in a superposition state, so you can easily see it could be a qubit. Whatever the case may be, here this structure is about 5 nanometers across, so these are not nanometer size coding elements, and that gives you a huge density of information storage. And that shows you all kinds of patterns that can emerge and illustrates the enormous potential of memory storage. And imagine this is just one patch, and there's thousands of patches on each microtubule, and there are thousands of microtubules in a neuron, and there are hundreds of billions of neurons in the brain. So if that's how it works, it's magnificently powerful. So that's the reference to this paper. We call it Brain's Memory Code. And here you see actually this enzyme, CAMK2 enzyme, walking on this. This is a cross-section of microtubule walking, and you can imagine extending a microtubule as a cylinder. So that would be that this enzyme is walking on it. As it walks, it phosphorylates various sites depending on what kind of information needs to be encoded here. So yeah, this is leading us to this more speculative aspect. Is there quantum computation taking place? And of course, it could, as I mentioned to you, it could be either as a result of tryptophan excitations, which are naturally quantum because it's photonic stimulated electronic transitions by photons, or it could be phosphorylation where phosphate groups are in a superposition state. Okay, so maybe I'll skip this. I'll just say that what I told you about Travis and myself, our publications is still pretty down to earth because the Orkawa hypothesis goes really far out, and it talks about these quantum states being present in a curved space-time, as in gravitational general relativity theory, and it can operate at all kinds of frequencies and different subatomic scales. It reaches to an incredible depth. I think it's, I would say, at the moment, very premature to claim that that's valid, but it's, yeah, speculative. So, yeah, this is kind of a summary where computational capacity of the brain may take us depending on various assumptions. And I will just stop here and thank you for your attention, and ask you if you have any questions about this.
Yeah, I'm interested in your thoughts on Michael Levin's bow tie idea. He describes this in great detail many times. What he's saying is what we remember is a very abstract, compressed, and time-limited portion of whatever happened to us. So that's the narrow part of the bow tie. You have your life experience, which is wide, then it narrows down into the memories, which are very compressed and maybe partial. And you can also wonder about the difference if you're remembering a symphony and if you're a, musician who actually plays it from memory. Maybe that memory is a lot different from what you or I would remember from the
I see.
same symphony. But that bow tie metaphor comes
Hmm.
up over and over again. And he seems very, very convinced that we never remember the entire thing. It's some very abstracted
Yes.
form.
Okay, well, it's very interesting, but I would say the following. I think this is yeah. So we never remember. I would interpret as we can never recall. It doesn't mean that it's not encoded. It is encoded maybe deeply, and it's not so easy to recover this memory or recall it. And but at the same time, as you know, hypnosis can bring back memories which
Mhm.
normally you wouldn't be able to express.
Right.
So perhaps they are deeply stored somewhere. And under hypnosis, perhaps it's a more pure, let's say, state of the brain without all the noise and interruptions. You can untie the bow tie, so to speak.
Yeah.
So yeah, it could make sense. I think it's possible. Oh yeah, and Sheldrake's field morphic fields, I think is the name. So there are these fields around us. I you know this is something again speculative, but I've been thinking a lot about the possibility of the information field. So it's not just in
Yes.
our brains, but there could be, let's say, information field in all of the space around us, some form of information. On a personal level, I can tell you that sometimes when you're in, let's say, historical buildings or places like that,
Mhm.
that saw or experienced some events, you kind of get the aura or some sort of effect on. Perhaps it's just imagination, but I would venture to say that there is some stored memory of past events, um, even in inanimate structures. So, any other questions? Guess not. It seems like
Well, I don't know. Maybe we should probe a little bit more. The four of you who stuck it out,
Uh,
Barbara. Yeah.
yeah, I wanted to ask about the movement of the cam.
Cam
Uh,
K2.
yes, on the microtubule. It's
Yes.
just
So
like
it's
straight or can go diagonally.
uh, good
So,
question.
like
Yes.
following the charges, always following the charges, but can go diagonally.
So okay, this kind of opens not a Pandora's box, but another line of investigation, which is
Good.
how motor proteins move on microtubules. And first of all, this is fueled by ATP. So
Yeah.
they use ATP for detachment from the surface,
Mhm.
including kinesins and calmodulin kinase. These are ATP driven motors. And I don't know specifically about CAMK2, but kinesins move in a processive manner, which means largely, almost exclusively straight.
Mm.
However, sometimes they can take a step to the side. It's a lower probability. And sometimes they can even go in a helical manner around.
Yeah.
And and sometimes they can detach spontaneously. So it's a complex answer to your question. All possibilities can be seen or have been seen. Um, and yeah, so I guess you're wondering about how to predictably phosphorylate sites with some meaning for the information
Yeah.
encoded, right? So it could be, but it could be achieved by several or a huge number of these CAMKey. I don't know what the number is, but let's say you unleash thousands of them, and then one will go along the straight line, another maybe in a helical manner around the microtubule, and so on. So they can, in fact, it's possibly how shall I put it? A nanotechnology, a nanobiotechnology device you could construct,
Yeah.
or even a computational device, maybe some nanocomputer based on these structures. Perhaps that should be patented.
Can they do a bridge together? Do you
Can they
think they
bridge
can?
between two?
Yeah.
Possibly. Well, from the point of view of distances, that's actually in this paper we speculated. In fact, you're right, because we speculated that imagine not a one microtubule, but in neurons there's bundles. Let's say they're separated by 90 nanometers, so it depends on the diameter of the dendrite, but could be, I don't know, 10 or 20 microtubules in a bundle, and they are interconnected by these maps, map tau. So these CAMK2 could move from one microtubule to the next on the map. And maps can be damaged or removed by pathologies, or in fact, in Alzheimer's disease, map tau is hyperphosphorylated. So instead of microtubule being phosphorylated, map tau is hyperphosphorylated, and that, what does it mean? It introduces extra negative charges. And remember, tubulin is negatively charged, so
Yeah.
negative charges repel, and then map tau would detach, fall off, and actually accumulate separately in these neurofibrillary tangles. The bottom line is, this process that you asked, is it possible, would stop, because CAMK2 would not, it would be probably too far for most of them to jump from one microtubule to the next, as they normally would go on tau. So this maybe is a mechanistic explanation of how memory loss occurs in Alzheimer's or cognitive decline.
Yeah, I think if I remember correctly, it's how also anesthesia it's also kind of explained because calm modeling is also in action during anesthesia. So
That's one, yes, one possible. I mean, anesthetic molecule depends on which ones and all
yeah.
this. They do all kinds of things. This might be one of them, preventing calmodulin kinase from binding to microtubules. Another one is what Stuart Hameroff again is believing that anesthetic molecules bind to tubulin and prevent these quantum excitations from occurring if it's a quantum memory. And then, of course, the more pedestrian approaches are that anesthetic molecules bind to the receptors on the membrane, and they prevent signaling across the membrane to take place. And one of my kind of expert advisors on these topics, neurophysiology, Bruce McIver, a Canadian from Calgary, but professor at Stanford, he calls it sufficient but not necessary. In other words, all of these things may occur, but they are not necessary. You can always find a different explanation for different types of anesthetic or different types of mechanism in anesthesia. So yes, all of it makes sense, but we don't have a single model of anesthesia yet, or explanation.
Yeah.
Years later, all that stuff. So that's another topic for another lecture, maybe.
Great.
So.
Okay, well, that was a wonderful lecture. The other students, do you have any questions? Going. Okay.
There's
All
always
right.
email. There's always email.
Yeah, there is. You can
If
email
students
Jack
are
if
shy
something
or maybe thinking
comes
about
to
something
you.
else,
Yeah.
multitasking. So, quantum brain theory makes sense, right? Because we can do all kinds of things at the same time, right?
Okay, great. Thank you very much. And we look
Thank you, and see you
forward to
back in Edmonton.
next term. Yeah. Okay. Bye-bye.
Oh yeah, before
Yeah.
no, I want to meet with you before next time to make a movie with Barbara in the
Yes,
lab.
sure. No, that would
So
be
that'll be
great.
probably in April.
Yes, I would be happy to do that.
Thank
Yeah.
you so much in advance.
Bye.
Okay, great. Bye-bye now.
Bye-bye.
Yeah.
Take care.